Hi, I'm Live. I'm Sarah Nikiforou, Medical Director of Dana-Farber Cell Manipulation Core Facility. And come along, let's give you a tour. In terms of our physical plant, we are one of the larger academic manufacturing facilities, and we supply over 1,500 products per year, primarily to Dana-Farber patients in the adult and pediatric clinics. However, we are also a core Dana-Farber Harvard Cancer Center facility. As such, we help supply products throughout the Harvard infrastructure in Boston area. And as the cell therapy field grows, we increasingly help out clinical sites around the nation, even for non-oncology indications, which cell therapy is making inroads into. So this beehive of activity is our QC testing lab. So here we will do things such as, once products are sampled upstairs, they will come down and cell counts will be obtained. These big gray boxes are flow cytometry machines, similar to what I mentioned, the Tito with the antibodies and the fluorescent molecules to be able to characterize cells. But here all we're doing is characterization, not sorting. But here's where we enumerate our CD34 positive stem cells to tell if someone has collected enough from their donor. We enumerate T cells for donor lymphocyte infusions, and then we have complex flow panels based on specific research needs. So for instance, projects involving cytokine-induced memory-like NK cells. So for that, we needed to design a panel that highlighted natural killer cells, was able to tell us how many contaminating, potentially alloreactive T cells there were, but also characterize the NK cells as to whether they were activated by cytokines or not. So that work happens here in terms of the development of the testing assays, as well as testing at various stages of processing themselves. So really almost everything we can do in-house or with our Harvard collaborators, which is why this is such a wonderful place to work. So this remains a vibrant, critical part of our laboratory. So our bread and butter, so to speak, historically for any cell processing lab has been processing of autologous and allogeneic stem cells for lymphoma and myeloma patients in high-dose chemotherapy and autologous stem cell rescue, and then primarily for leukemia patients in allogeneic transplants. And behind me, you can see one of our two large open cores with 6 workstations in each room. And each workstation consists of a biosafety cabinet, a refrigerator, and a centrifuge. Each of these products, no matter what type, will get sampled for cell counts, for viability. We will characterize the types of cells that are in that particular product, such as stem cells or T cells, and we'll sample for sterility to know it's safe to get back to the patient. In addition to the standard workflows, more complex processing, such as depletion of TCR alpha beta positive cells or even naive T cells, can be accomplished to eliminate the cells primarily responsible for acute and chronic graft versus host disease. We can also positively pull down cells like regulatory T cells and give them back to patients to treat or prevent graft versus host disease. This open core and this staff is totally ready to perform those selection or depletion processes. The other thing that happens up here, and as you scan around our receipt and distribution room, is what we call cell pharmacy. Meaning that as cell therapy has become less of a niche transplant center field and much more a commercial entity, increasingly one of our roles is to obtain raw material from the patient's bloodstream usually, to make sure it is properly labeled and to send it out to these commercial manufacturers. Then when it comes back, we also have to check the proper labeling. So there is a huge emphasis in the field And this entire room where I'm standing is devoted to making sure that all the documents, all the labels properly track chain of custody and chain of identity. So I mentioned previously that the novel cell therapy side of the lab does the more complex genetic manipulation and will culture things overnight. One of the beauties in here, in our facility under Dr. Ritz's initial guidance, was that both the more standard of care and the more novel processes were combined into one manufacturing group. That means that all of our core infrastructure with QA and QC testing for et cetera, is shared, right? And so that any one development in one part of the lab can be cross-pollinated into the other. So unlike some academic facilities where you have individual labs with individual areas of expertise, we bring it all together. Down here and in this room, you can see they're designed totally differently than our open cores. So this hallway down here, which is clean, controlled airspace, has individual small rooms over on the left-hand side with an airlock. Most of them are negative pressure, so we're not blowing out genetic material that we're manipulating. And they will have whatever equipment you need, because there are various platforms on which you can do genetic engineering. This is an example also on the NCT side of a room that is designed differently and really would be used for only one thing at a time. That is a Tyto machine, also made by Miltenyi. But what this allows us to do is, instead of using antibodies and iron beads plus a magnet to pull out, yes, no, a cell population, this allows us to use regular flow cytometry antibody and fluorescent molecules to sort on multiple aspects of a cell of interest at once. And flip will allow you to sort individual cells and get extremely high purity. So this is actually a platform and a process that we have just gotten FDA approval for under Leslie Kean to be able to isolate regulatory T cells, expand them outside the body, and give them back to children who have graft versus host disease post-transplant. Now you're up. Okay, and this is one in which we do genetic manipulation. So we actually have, although there are many platforms, our genetic manipulation typically for chimeric antigen receptor T cells will happen on the Miltenyi Prodigy machine, where you're able to select T cells, put them directly in a closed system into a culture chamber, control the media, the cytokines, whatever viral vector in most cases you're putting in, and in a semi-automated way, get a CAR T cell within 5 to 7 days, and increasingly that time is being shortened. So that is something that we do here. This is the platform we use, and this is an example of one of the rooms that we would utilize this in. We also do genetic manipulation, not just of T cells, which is what the CAR T cells are most commonly used, but of NK or natural killer cells, and also stem cells, where we select for CD34 cells first. And we also have a genetic gene editing platform using CRISPR-Cas9 that we are open and enrolling to that trial. We have the flexibility to be able to modify our manufacturing, to be able to modify our QC testing. Obviously, we have to tell the FDA and make sure it's okay. However, that's one of the things that we are able to add, and we have incredible subject matter experts, potentially including myself, who look over every single aspect of manufacturing and of in-process testing to be able to feed back to the sponsor in real time when things are working or not. The one place I'm not going to have time to show you is what we call our process development lab. So you can imagine that there is a lot of work that goes in to getting things ready from benchtop to being able to go into patients, especially under the rigorous regulatory requirements that we have. So we offer process development services, and this was a new group of the lab that we added within the past few years to be able to help basic researchers bridge that gap so that what comes into us in the facilities that you've seen are really things that are ready to be validated for INDs and get into patients. So in closing, hopefully I've given you a flavor of the processing spaces that we have within the DFCI CMCF that are similar to some of the other manufacturers on the commercial side or in larger academic centers like us. Originally, this was really for hematologic malignancy patients. We are now manufacturing for neuroblastoma, like Ravi Mazer's trials, and Dr. Rami's trials. We are targeting head and neck cancer, ovarian cancer, renal cell cancer, right? So that's growing. And then I mentioned increasing the non-oncology indications, which is really exciting to meet with our rheumatologists, our neurologists, and we're branching out in different clinical directions. And our clinicians may be based at Dana-Farber or in the Harvard Cancer Center, or even across the nation, because as long as we have an infrastructure that supports our regular mission, we're happy to ship all over. OncLive, thank you for asking the CMCF to contribute to your readership and hopefully their enjoyment. And I'll be handing you off now. Thank you.